Hi everyone, I'm going to talk about bacteria and viruses. First, let's talk about bacteria. Bacteria is a prokaryotic cell, which means that it doesn't have a nucleus, so it has one circular chromosome-free in cytoplasm. It has ribosomes, it has a cell membrane, it has a cell wall, and some bacteria secrete a layer of polysaccharides around the cell wall, forming a capsule. On the outer surface of some bacteria, there is a hair-like structure called pili to help the cell attach to surfaces and to help in conjugation, where plasmids are transferred from one bacterium to another, and this leads to variety. Bacteria also has flagella, which helps in movement. Bacteria are typically 1 to 10 micrometers long and 0.7 to 1.5 micrometers wide. There are three main shapes of bacteria. Round bacteria are called cocci, rod-shaped bacteria are called bacilli, and spiral-shaped bacteria are called spirochetes. Only a small percentage of bacteria cause disease. So how do they cause the disease? Some bacteria multiply quickly at the site of infection, then they might spread to other parts of the body. Other bacteria secrete a toxin which causes harm. Some common bacterial diseases are strep throat, acne, cholera, and many types of food poisoning. Now let's talk about viruses. Viruses are considered non-living, as they lack some characteristics of life. Most biologists don't consider viruses to be living, as viruses have no organelles to take in nutrients or use energy, they cannot make proteins, they cannot move, and they cannot replicate on their own. A virus has genetic material within a protein coat. Some types of viruses have an envelope and spikes. Most viruses range in size from 5 to 300 nanometers. So this is a comparison between a bacteriophage virus, which is 225 nanometers, and a bacterium, which is 2 micrometers. Viruses can be spherical, polyhedral, helical, or complex. To replicate, a virus must enter a host cell. The virus replicates by either the lytic cycle or the lysogenic cycle. In the lytic cycle, the virus attaches to the host cell using specific receptors in the plasma membrane of the host. Then the genetic material of the virus enters the cytoplasm of the host. The virus then uses the host cell to replicate. The viral genes instruct the host cell to make more viral capsids and enzymes needed for replication. The capsids form around the nucleic acid of new viruses. The new viruses leave the cell by exocytosis or by causing the cell to burst, releasing new viruses that are free to infect other cells. Viruses that replicate by the lytic cycle produce active infections in which the symptoms start to appear 1 to 4 days after exposure. Now let's talk about the lysogenic cycle. In the lysogenic cycle, the virus attaches to the host cell, inserts its DNA, and the DNA enters the nucleus of the host cell and integrates into a chromosome. Once integrated, the infected cell will have the viral genes permanently. The viral genes might remain dormant for months or years, until it is activated by a trigger. Activation results in the lytic cycle, and the new viruses are released, free to infect other cells. There is another type of viruses which has a complex replication cycle. This type is called retroviruses. A retrovirus is a virus that has RNA as a genetic material. This type has a complex replication cycle compared to the viruses with DNA. First, the virus attaches to the host cell. Then it inserts its RNA. Then the enzyme, reverse transcriptase, synthesizes DNA using the viral RNA as a template. And the DNA integrates into a chromosome in the host cell nucleus and lies inactive. Once it is activated, RNA is transcribed from the viral DNA, and the host manufactures new viruses to complete the lytic cycle. Some common viral diseases are common cold, influenza, rabies, chickenpox, and warts. That's all. Thanks for watching.